Morning students. Today we are discussing a topic called excretion. Excretory system is the third unit of your syllabus. The unit includes a brief account of excretory organelles in different animals. That is in contractile vacuole, protonephridia, nephridia, Malpighian tubules, antennary glands, kidneys, etc. These are the different excretory organs present in animal kingdom. Okay. After that, we are discussing different types of kidneys, pronephros, mesonephros, metanephros, etc. After that, we are discussing different type of excretion, ammonotelism, uricotelism, ureotelism, etc. And urea cycle also. Urea cycle is the cycle of formation of urea. And another topic which comes under this unit is mechanism of urine formation in man. Okay. So these are the topics we are going to discuss in this unit. First of all, we are discussing what is excretion. All of you know what is excretion. Excretion is the process of removal of waste products which are formed after metabolism in our body, and the process of removal of these waste products is called the excretion. And excretion and osmoregulation are interconnected process, and these two are the main functions of any excretory organ present in any organism. Okay, through excretion, organism controls osmotic pressure. How they controls the osmotic pressure? By controlling the ionic balance, that is balance between organic ions and water, and also maintain acid base balance in our body. Thus, excretion promotes homeostasis and a constancy of organisms internal environment. Now we are going to discuss different excretory organelles present in different group of organisms. We are starting from very primitive organisms like protozoans. All of you know in protozoan excretion take place by using organelle which is called contractile vacuole. Okay, contractile vacuoles or regulatory organelle which are usually spherical in shape, found in freshwater protozoa and lower metazoans such as sponges and hydrats, that collects excess fluid from the protoplasm and periodically empties it into the surrounding medium. We know these organisms are found in water, so excess water may enter into their body. Then contractile vacuoles collect this water inside this contractile vacuole, collect this water, and they empty this water out of their body. Okay, thus they maintain a constant internal environment inside the organism's body. Otherwise, our organism may burst out due to excess water. It may also excrete some nitrogenous waste. We also know every organism there will be some metabolic process taking place inside organism's body. So after that metabolism, different nitrogenous waste materials are formed. Okay, then this contractile vacuoles also collect nitrogenous waste materials from the body of these organisms, and along with water they empty these things outside organism's body. In amoeba, it changes position with the animal's movement. That is, the position of the contractile vacuole changes according to the movement of the organism. In most ciliate, it follows a definite path through the cell, but in some organisms they follow a definite path. In euglena and other flagellate, it remains stationary. Euglena is another protozoan. In euglena, the position of the contractile vacuole is permanent. That means it remains stationary in euglena's body. But we already discussed that in amoeba it is moving. It is constantly moving according to the movement of the organism. Filling and emptying cycle may be last for few seconds or some minutes depending upon the species. The time taken for this process may vary. It may be few seconds or maybe some minutes. Okay. You can see the picture of contractile vacuoles which is present inside an organism, paramecium. Here in this slide you can see the picture of contractile vacuole, and another picture also. Here you can see water enters into the body of amoeba. When water is excess, which is collected into the contractile vacuole, then contractile vacuole swells. When it fills, it completely fills, it moves towards the edge, towards the plasma membrane, and it bursts out and removes water and other waste product outside of the organism's body. Nephridium is the another structures which is found in many invertebrate organisms, which functions as their kidney. The nephridium is an invertebrate kidney organ found in pairs and performing a function similar to the vertebrate kidney. Nephridia removes metabolic waste from animal's body. Okay, there are two type of nephridia, protonephridia and metanephridia. You may be knowing it. What is the difference between protonephridia and metanephridia? In this picture, you can see, in this slide, you can see picture of two kinds of nephridia, that is protonephridia and metanephridia. What is the difference between these two? In protonephridia, you can see one end is blended and only one opening is present. Okay, it is nephridio 4. But in metanephridia, two openings are there. One opens into the body, inside the body of the organism, and another opening is towards outside the body of organism. Okay, the opening which is present inside the body of the organism is called nephrostome, and which opens outside is through nephridio 4. This is the main difference between protonephridia and metanephridia. So protonephridia, we already discussed that there are two types of nephridia, protonephridia and metanephridia. Okay, protonephridium is a network of tubules found in different phylums like Platyhelminthes, Nematia, Rotifers, etc. Okay, one end of this protonephridia will be blind, that which consists of a special type of cells which are called flame cells or solenocytes. In some organism, inside the cell, cilia are present, a tuft of cilia are present. Then that kind of cells are called flame cells. End cells are called flame cells. And in some other organism, the cell contains a tuft of flagella. In that organism, the structure is called solenocytes. Okay, perform the function of excretion as well as osmoregulation. Okay, protonephridia are made up of many flame cells. In flame cells, there are, I already told you, there will be a tuft of cilia. Okay, the backward and forward movement of this cilia creates a vacuum inside this flame cells. Due to this, the material from the body fluid enter into the flame cells. That is the different kinds of material that may be needed for the body, or some of the material which are waste product. Okay, both of these materials are all these materials are enter into the flame cells, and when it moves through the tubules of protonephridium, the things which are needed for the organisms are reabsorbed back into the blood. The remaining material is excreted outside of the organism through an opening which is called nephridiophore. So, in this picture, you can see the structure of protonephridium. This is the tubule and opening nephridiopore, which opens outside to the organism's body. One of its end contains a special type of cells which is called flame cell. You can see a tuft of cilia inside this flame cell. Next, metanephridium. A metanephridium, it's a type of excretory gland found in many types of invertebrates such as annelids, arthropods and mollusca. That is organism or a group of organism in higher than this platyhelminthes and rotifers. Another type of nephridium is present which is called metanephridium. I already told you the two end of this tubule will be opened. Okay, a metanephridium typically consists of ciliate funnel opening into the body cavity or coelom, which is called a nephrostome. The one of it, I already told you, two ends are opened. Okay, one of its end, it consists of a funnel like structure which opens into the body cavity or body fluid of the organism, which is called nephrostome. The opening is called nephrostome. It's a funnel like structure. Connected to that, there will be a duct with many variously glandularized, folded and expanded. That is, after nephrostome, you can see a coiled tubule. Okay, this coiled these tubules are ciliated. Okay, which typically opens to the organism's exterior by an opening called nephridiopore. So nephridiostome, which is the which opens into the body fluid, and another opening which is called nephridiopore, which opens out of the organism. Okay, the waste materials or from the blood material enter into the funnel like structure called nephrostome by the beating of the cilia. When it moves through this coiled tubule, the important materials are absorbed back into the blood, and remaining materials are remaining waste product or waste material is excreted through nephridiopore to the outside of the organism. Okay, I already told you these ciliated tubules pump water carrying surplus ions, metabolic waste, toxins from the blood and useless hormones out of the organism by directing them down funnel-shaped bodies called nephrostome. That is the cilia, which pump water which contains ions, metabolic waste, toxins, food and useless hormones from the body fluid to the funnel shaped nephrostome. Okay, this waste material is passed out of the organism, I already told you, through nephridiopore. The primary urine produced by the filtration of blood is modified into secondary urine when it moves through this coiled tubule by a process called selective reabsorption. Okay. In this picture, you can see a structure of a metanephridium. It has two opening, nephrostome and nephridiopore. Okay, the cilia, you can also see the cilia here. The beating of the cilia cause the movement of material into the nephrostome, and it travels through this coiled tube, and finally the end product comes out through external opening called nephridiopore. Another structure which is found in animal kingdom is Malpighian tubules. Okay, this Malpighian tubule system is a type of excretory and osmoregulatory system found in some insects, myriapods, arachnids and tardigrades. The system consists of branching tubules extending from the alimentary canal that absorbs solute water waste from the surrounding medium. That is, Malpighian tubules, a group of tubules which are present in the alimentary canal of the organism, which extend towards the body, inside the body, which extends from the alimentary canal to the exterior of the body or body fluid, and which absorbs solute water and waste material from the surrounding hemolymph. In insects, any of the excretory organs that lie in the abdominal body cavity and empty into the junction between midgut and hindgut. In species having few Malpighian tubules, there in some species the number of Malpighian tubules will be very less. In this case, these tubules will be long and coiled. But in some other species, numerous tubules are present. But in this case, the tubules will be short. Okay, the tubule cells actively transport initial urine constituents, that is potassium ions, water, urate ion, sugar, amino acid into the tubule. Okay, later the water and nutrients are reabsorbed back and final urine is formed. In some species, urine is acidified into the distal end of the tubule and an aqueous suspension of uric acid crystal is conducted into the rectum. It's a picture of honeybee. In this you can see the structure of Malpighian tubules where it is located. It is located in the junction of hindgut and midgut and hindgut. You can see many tubules in this picture. It's also a picture of the small pigeon tubules. You can see a section of alimentary canal in this picture where it is located. It is the junction between hindgut and midgut. Many tubules extends towards the body coelom or which are present in the body fluid. Okay, these tubules absorb salt, water, and another nitrogenous waste materials. Then it comes into the alimentary canal from where important things are reabsorbed back, useful things are reabsorbed back into the hemolymph, and remaining waste material passes out from the body as feces and urine through rectum. So another structure is antennary glands or green glands. In decapod crustaceans, excretory organs are known as antennary glands or green glands. You studied the structure of you already studied the structure of antennary glands in prawns. Okay, these glands are opaque white pea-seed sized structures enclosed in the coxa or second antenna, which is located in the coxa or second antenna. So they are called antennary glands. Each gland has three parts, the end sac, labyrinth and bladder. Each antennary gland consists of three parts, the end sac, labyrinth and bladder. The antennary glands are complex nephridia-like structure and they extract nitrogenous waste product and excess water from the blood in the same manner as the vertebrate kidneys do. These glands also perform the function of osmoregulation. I already told you three structure are there in a antennary gland. Three structure are there: end sac, labyrinth, and urinary bladder. The end sac and the labyrinth are the two regions which are responsible for extracting urine from the blood. The most nitrogenous products include ammonia, a major excretory product in all crustaceans, and other ammonia compounds are excreted by end sac. The other excretory products are conveyed by the excretory duct of the labyrinth from the surrounding blood of the hemocoel. The labyrinth was folded and glandular, which are considered as the site of selective reabsorption. That is, reabsorption of the material from the primary urine formed inside the labyrinth is take place when it moves through the urinary tubules. The primary urine, that is, the primary urine is modified when it passes through the parts of excretory system. The urine remains temporarily stored within the bladder and it periodically expelled through renal pore. In this picture, you can see the structure of an antennary gland. It has an end sac, a spongy labyrinth, and excretory tubules which opens into a urinary bladder. Okay, when the primary urine moves through this excretory tubules, the selective reabsorption of the material take place, remaining things, that is the final urine which is stored inside this bladder and periodically expelled out through this renal pore. So these are the different types of excretory organs present in the different group of organism. And thank you for listening. Thank you.